буквальном, оказывается, смысле. Спасибо, и я сейчас, у нас еще один доклад. Я приглашаю на сцену Рэндала Коene. Тема выступления «Инженерный вызов. Как сделать наш разум субстрат независимым через эмуляцию мозга уже в течение нашей жизни». Да, окей, Рэндал, it's your time now.
Hi. So, my name is Randal Koene, and part of my work, part of the job that I do, is that I work with a company called Halcyon Molecular to build basically the world's most accurate and long read length genome sequencing, which is something that we need people with that have lots of different skills. And my background, for example, is physics and electrical engineering, information theory, computational neuroscience, and neural engineering. But in addition to that, what I do is I am the founder and organizer of a sim group, which I'll explain in a second, called Carbon Copies. What I'm going to try to explain today is what that group does, which is to bring together the experts and projects needed to do something called substrate-independent minds. And I want to tell you what that is, and why it's important, and how it can be done. Specifically, I want to talk to you about how feasible it is. So, in groups like this, we often talk about things like life extension or life expansion, augmentation, matters like that. And when we do, I think it's important to try to consider what the objective really is. What is it that we're trying to do? What I mean is, what are we trying to extend or augment? And so I want to take a little step back and think, what are we? What are you? If you look at these slides, you can see in the background a lot of different experiences a person can have. So experiences are a part of you. Another part of you is your body. It's the senses that you have. It's the actions that you can take. And then on the right, you see all these different expressions. I'm trying to represent the idea that each one of us has unique characteristic responses to things. So if you put us in the same situation, with the same input, a different individual reacts slightly differently. There are unique responses that we have. So where does all of this actually take place? Now, all of it takes place in the mind. That's where we do all of this stuff. Really, I can't even touch anything in the universe. That's how unreal that is. Imagine that I'm trying to touch this table. If you think about the atoms in my finger and the atoms in the table, the protons and neutrons in there, they never actually collide. It's mostly space. There are some forces there. There's electrical activity that goes up my arm, ends up in the brain, and then I don't notice anything. I don't realize that until something's being processed. So, when we talk about things like life extension, what we really mean is that we're trying to safeguard those processes, that being, that experience of being that's going on inside our minds. And there are really two different ways that you can do it. One is that you can say, we know that right now all of this processing is taking place in the brain. So what you need to do is you need to maintain the brain. You need to make sure it keeps functioning well. You need to keep the body functioning well so that it keeps the brain going. And you need to make sure that your environment stays okay because the body depends on that environment. It's not built for every possible environment. Imagine if we suddenly lost the atmosphere. Well, this body would die and the brain would go and everything would stop. But we also just said that what we're really talking about is the processes going on in the mind. That's the experience of being. So the alternative is that you could address that directly. Do what you would do if you were treating it like a valuable piece of information or valuable program. What would you do with a valuable program? The first thing you would do is that you would make a backup. You would archive something. The next thing that you might want to do is that you want to be able to fix errors if they're there, or you may want to be able to upgrade it to run on the next best hardware. So you want access to the code. That's really what I'm talking about is about. Substrate-independent minds is about data acquisition. It's about access. The notion being that right now these processes in my mind are running on a kind of substrate. But if they can run on many different kinds of substrates, then we call it substrate independent. If by analogy, think of computer programs that are platform independent code. They can run on different kinds of platforms. Which of these two strategies is better? Well, I think they're both valuable, and they should be looked at very concretely. If you're thinking, where would I put resources, where would I put effort, what can we achieve within our lifetimes, then you need to look very concretely at the plans that are there. So that's what I hope to do today. But before I rush into that, just a quick 2 terms. What you hear a lot of people talking about in some communities when they talk about technologies like this is the term mind uploading. Mind uploading is a perfectly good term, but it doesn't say anything about what you do once you put your data somewhere, once you upload it. So it's not very objective oriented. It's really about the transition from this biological substrate to some other one. And then where would you like to end up? If you're trying to do substrate independent minds, there are many ways of doing it. And preferably, if this were an ideal case, on every substrate you would go to, the code would be optimized for that. It's like compiling a program. But we don't really understand it that well. If you ask a neuroscientist, do you understand this part of the brain? They will always say no, because they know that what you're actually asking is, do you understand all the different strategies that the brain is using at different levels of the hierarchy, right down to the neurons? And of course we don't. So we can't use that approach right now. But what we can do is we can go down to the bottom level where we know more about how neurons work and how they pass electrical information off to one another. And this is what we call whole brain emulation. The idea that you can emulate the neuroanatomy and the neurophysiology at that level. Okay. Oh, how did I do that? Okay, let's get back down here. Now, this isn't an entirely new field. I know it hasn't been talked about as much as maybe biological methods, like when you hear Aubrey de Grey talking about SENS Foundation and things like that. But there has been work going on at least since 1994 by researchers dedicated to the pursuit. There was the Mind Uploading Research Group that I inherited from Joe Strout, who's the person in gray up there. The knife-edge scanning microscope was developed by Bruce McCormick, who's right next to him. And then in 2007, we had an Oxford workshop about whole brain emulation that a roadmap came out of, published by Anders Sandberg, who you see in the picture down here next to Suzanne Gildert and myself. And then many others, interesting people like Ted Berger and Ken Hayworth, Winfried Denk, Sebastian Sung, who popularized the idea of the connectome, all the connections of the brain being important, at the 2008 Neuroscience Conference, Peter Passaro, Yoon-Suk Cho, Jeff Lichtman, Diana Deca, and then of course we have the formation of carbon copies down there with Suzanne Gildert and myself. And since then, it's become much more mainstream. We have people like optogenetic star Ed Boyden working on brain emulation and extracting circuitry for neural circuit function emulation, and efforts by, for instance, IARPA interested in our work. Now, if you want to do substrate-independent minds, there are different routes you can take, and I'm only going to talk about one today. Another really interesting one, besides the whole brain emulation route, is the brain-computer interfaces route, but it has a lot of other driving factors behind it because it has a lot of commercial potential at every stage. So it was so important that I thought this year in 2012 we'll just do a whole separate workshop on that. So I'm leaving that for then. Now, if you're going to try to do something like whole brain emulation, then you need to make representations of things. And every time you make a model or a representation, you need to choose a resolution, some level at which you're going to describe the elements and describe what they do. That means you need to characterize them. And when you're characterizing them at a resolution, it means that below that level, below that resolution, at any higher level, you don't talk about the mechanisms that are going on inside. Instead, what you're doing is you're looking at their input and their output, treating it like a black box and describing what the functions are there. And you want to capture all of them. You don't want to miss any latent function. And in these boxes here are just some examples. In the top, you see how Wu et al. Did that for sensory neurons. In the bottom, you see the famous Eugene Itzkevich neuron model, where it uses several parameters to easily model many different biological types of neurons. And on the right is some work of my own, where I focused mostly on the currency of the brain, which is the spikes in the system. After all, when a neuron spikes, and the exact timing of that spike is both what determines where memories are laid down, so what things are remembered, as well as which muscles are activated, so how we can interact with the environment around us, even just by speaking. So these are important issues if you want to model something correctly. Then above that level, the important thing is to capture emergent properties. So you have these elements, you characterize them, and what you need to do is you need to understand how they all work together, because this gives you emergent function. And a good way to find out what they're doing together is if, first of all, you know how they're connected. That's why extracting the human connectome, all the connections in the brain, is really important. Now, you can do this black box selection, which level you want to work at, in many different ways. And in SIM, this happens depending on the route people choose. There's a route called loosely coupled offloading, or at least that's what we call it generally speaking, where the entire body or person is considered a black box. And what you're trying to do is model their behavior, model how they act, so that you could basically make a simulacrum of them and say, this is that person. What it needs is you need video recordings, audio recordings, self-report, maybe an AI that learns how to interpret what you're doing. This is very similar to a method called the Bainbridge-Rothblatt model that is used for trying to create what they call an upload. Now, there are differing opinions about that. There's also, at the next level, you can take the entire brain or parts of the brain as the black box. This happens, for example, when you have a cognitive architecture that you find an interesting or correct architecture for the human brain, and you say, this area does that, that area does that, that area does that, and you want to model how each one of them works and maybe personalize it so that it seems like one specific person. But then, at the next level, this is where it gets really interesting. You can take either neurons or parts of neurons, like the morphology of neurons, as the black boxes. And this is what's done in computational neuroscience and neuroinformatics. And it's the level that I'm going to be talking about because it's the one that's the most concrete and most usable right now for whole brain emulation. They're also interesting for brain-computer interfaces, of course. Now, if you're going to do whole brain emulation and you look at it as a strategy, as a roadmap, there are really four big requirements, four things that you need to do. One of them, you choose your resolution and scope, as I just said. You need to validate that. You need to do some tests on that. I know a few people who are, and it's very interesting work. You also need to extract that connectome, the structural connectome. Where are the things connected, as I was just saying? And because you need to characterize all of those elements that you chose at that bottom resolution throughout that connectome, basically you need to get a functional connectome. And finally, because you want to re-implement this, you need some sort of platform that can emulate. Okay, just a quick word on function and structure. That's something that often gets brought up about neural networks, and it's made to look rather mystical, but of course, function and structure and their interaction happens in every kind of system that we have. If you think, for example, about a computer chip, it's full of transistors. Those are the basic elements, and then they're arranged in a certain structure. Now, if we're looking at scope and resolution, then there are sometimes shortcuts that we like to try to take because it's a very difficult problem. So there are two different kinds of shortcuts that have been contemplated. One of them is look only at the function. Take neurons, record from the neurons, and if you record from many neurons at the same time, then instead of looking at how they're connected by actually looking at those connections, you can derive connectivity because you see how they interact. So if you use something like Granger causality, for example, then you can try to calculate a functional connectivity map. A problem with this is that you easily miss latent function, because if you want to study a very complex system that has a lot of elements inside of it, and you want to see everything that it may have remembered and how it can respond, you have to observe it over a very long period of time, perhaps since birth. Well, the other shortcut is to just look at structure. What you can do is you can look just at what is the morphology of a neuron, how does it look, and you've studied many neurons that look the same way, and you know that they generally have a certain kind of receptor channels, so they use certain neurotransmitters. You know that they respond in a certain way, so you can build a library where you can map from this morphology to that kind of function, and you can map to parameter distributions. And then if you have a very detailed morphological model, you can make what you see on the right there, which is a compartmental model where you build a very large model morphologically of what the neuron is like, and each of the little compartments is basically an electrical circuit model. You set the parameters according to what you found in your library, and then you hope that the entire system works. Now, the problem with this is that measurement is never quite precise, and also we need to understand that that library may not be a one-to-one mapping. Is there always just one morphology that maps to one type of function? So that may break down there, and in terms of errors, because when we talk about error in neural networks, one of the things they're famous for is that they deal well with error. If you have random errors, the system's fairly robust. But that's not true for systematic errors or for cumulative errors. And if you have electron microscopy, for example, you have areas that are out of focus or where you don't know exactly what your resolution is. So you may be measuring things wrong. The same when you're cutting slices of brain. You have a knife that has features in it. So you have characteristic errors, and those accumulate. Now, it can be very difficult to tune a large system like that. If you take the entire thing and you just try to tune it with hundreds of billions of 100 billion neurons in it, then this becomes a problem that is way too difficult as an optimization problem, even for quantum computers when they eventually come along. We can look at it, at least explore it, using something like this. This is a modeling environment that I originally built to try to explore what the emergent functions are that pop right out of the structure as neurons grow and connect into networks. But it can also be used to generate models where we know exactly what we're putting in terms of function and in terms of structure, so that if we can look at what kinds of errors tend to occur, you can find out how bad is it when you get these cumulative errors. But in the end, what you really need to do is simplify the problem. This is a problem of system identification. And what can you do in system identification to make it tractable, to make characterization possible, and to make the whole problem feasible computationally, is to make it a smaller problem, to simplify it. You want to take subsystems out of there where it's very easy to describe the input-output functions, where it's easy to get a characterization. That means you need to have those functional responses at that level. So this means you need high-resolution functional characterization, functional measurements, not just structure. And that's kind of what I'm trying to get to, which is we need both of those, structure and function. And there are real projects going on to do this for whole brain emulation. You see here the 4 yellow requirements. And on the bottom, I didn't put in the projects for the leftmost, but that's okay. Anyway, you see on the bottom a number of projects that are going on, and I'm only going to talk about 2 because of time constraints today, but they're all very interesting, so I'll quickly run through what they're about. If you want to get the structural connectome out, well, the obvious thing to do is to say this is structure, so it's spatial, it's something you want to look at. So what you can do is slice it really thin and look at the brain under an electron microscope and reconstruct. I'll talk a bit about that. But the alternative is that you say what we're really interested in is the connections and not all of this other messy stuff. So Anthony Zador at Cold Spring Harbor and Ed Callaway, they're working on methods using a virus basically to transfect neurons in mass and to deliver unique DNA barcodes that go to the presynaptic and postsynaptic sites. And then when you pull out sort of a biotin tag that's connecting the presynaptic and postsynaptic site, you pull out both the barcodes, and it's like pulling out pointers that are pointing to each other saying, this neuron's connected to that neuron, and this one's connected to this one. It's an interesting approach. Functionally, you also have a few different ways of going about things. There's a general idea of hierarchical measurement that we call the Demox tree for demultiplexing, and a specific implementation of that one is where Rudolfo Linas came up with the idea of making nanowires that you can push through the capillary system in the brain so that it reaches every neuron and can measure from them. The nanowires actually exist. They've been developed at the New York University School of Medicine, but there is still no way of actually getting them to branch like that. And also, they consume a lot of volume in the brain, which is a bit of a problem. On the other hand, you again have a biological approach, just like I was just saying about Anthony Zador. The good thing about biology is that you can easily apply it en masse in large amounts, and also it's already at the right scale. So there's this thing called the molecular ticker tape, which is a project that Ed Boyden at MIT and George Church at Harvard University, Conrad Korting at Northwestern, and Halcyon Molecular also working on, which is the idea that inside the neurons, you can record voltage events with voltage-gated channels, and you can affect the writing on a DNA being used as a medium for putting information on it, telling you when an event occurred. And then all you have to do is pluck out that DNA and sequence it, and you know when activity has been going on. So you can do this in many neurons at the same time. The problem with this approach is that biology is still kind of difficult for us to work with as an engineering project. There's a lot of random searching around for the right tools to use. What we'd really like to do is work at the cellular scale and be able to engineer to our heart's delight. And that's kind of the third one, which I'll talk about specifically. You see it already shown as a picture over there. Okay, and then lastly, yeah, it'd be nice if we can have hardware that's actually designed for the problem, so co-designed to what we're trying to emulate. So neuromorphic chips are a really good thing coming out. And there's just an example from the IBM Synapse project that they're doing for DARPA. Okay, so volume microscopy, or tape to SEM, which is the approach of actually looking at it to get the structure out. This is something that Winfried Denk has been doing in Germany for a while with something called SBF-SEM, that's serial block face scanning electron microscopy. So he takes a block of brain tissue, He takes an image, then ablates off a piece of the surface, another image goes all the way down. The problem with this approach is it doesn't work very well for large volumes. But Ken Hayworth has been working at Harvard for many years on building a system that can deal with the whole volume of the brain. And that's called tape to SEM, or it used to be called the Atlam Automatic Tape Collecting Lathe Ultramicrotome. The idea is that a diamond knife, cuts off pieces of this block of brain tissue and puts it on a tape. And the tape can be stored, as you see on the right over there, although it looks a bit messy. And you have random access to all of those pieces so that you can do microscopy on it all the time. Now, when you look at what these actually look like, you can see inside the red square here on one of these images, you can see where there's a synapse that's actually approaching a dendrite. So you've got that dark sort of area where the two meet, and you could reconstruct the morphology of them if you had many of these slices on top of one another. And you can even see little circles where the white arrow's pointing. Those circles are vesicles containing neurotransmitters, so you can even get an idea of what the chemical strength of the synapse is just by looking at this. And here you see an example of reconstructions like this, where you eventually get the whole cell. And with the help of Winfried Denk, Brigman et al. And Bock et al., they recently published 2 papers in Nature in 2011, in which they explored this method. And Brigman et al. Worked in the retina, Bock et al. Worked in visual cortex, in the visual system. And what they did is that, well, for example, Brigman, he first looked at the retinal cells functionally, observing how they operate, which receptive fields they have, so what they were sensitive for. And then he did the serial reconstruction, used that to predict what they would be sensitive for, and found that they could predict that function from the structure. So they verified that indeed this is possible, at least when you know a lot about the system. Okay, so now we'd like to be able to do something similar functionally, and something that we are rather good at is working with integrated circuit technology. That's something we have experience with. We know how to make hierarchies of systems that work, how to do communication, how to do aggregation of data, measurement, etc. When you actually look at what's possible right now with the resolution that's available in integrated circuit technology, if you want to get down to the cellular level and you make a circuit that's at the size of a red blood cell, so 8 micron, you can build something that can be powered, for example, by distributed infrared radiation. Infrared radiation at wavelengths between 800 and 1,000 nanometers is in what we call a transparency window for tissue, so it doesn't get absorbed by the tissue. So you could use that for communication as well as power. Also, if you want to do passive communication, it's like RFID, RFID tags that work with wireless frequencies. If you want to do this in tissue, again, you can go to infrared. And Yale McGuire at MIT is doing that specifically for these kinds of ICs that they want to use inside the body. So we've got both communication and power, and you can stuff about 2,300 transistors on something this size. That's the same number of transistors as in the original Intel 4004 CPU. And if you looked at today's technology, actually you can make that 4 times as much. So you can put as many transistors on there as were in the guidance system of the original cruise missiles. Now, you also need to make this work in the body, so it needs to be biocompatible. The easiest thing is you just stuff it in a little blob of silicon. But you could also encase it, for example, in artificial red blood cell, which is basically a protein shell that's been constructed, which can be functionalized so that you can use it in many different ways. But again, this isn't exactly everything we want. These hubs, they're complex, they can do data aggregation, they can do those sorts of tasks, but they're still fairly large. They work inside the vasculature, which leads to every neuron, but we'd really like to work outside the vasculature in the interstitial spaces between cells as well. For that, you need something smaller. But this is where it's nice that in integrated circuits, we can easily make these kinds of hierarchical systems, or teams. So you can make, for instance, 2 micron large ones, which have less transistor surface, but just enough intelligence to be able to do something simple, like sensing or stimulating when required, as long as they're in contact with such a hub.
And Randal, terribly sorry, but let me remind you about time.
Yes.
Yeah, horribly over time.
I will hurry through the end.
Yeah, okay. Thanks.
What you end up with is a cloud of computation, basically, that you can use in the brain concurrently with its activity in vivo. And the nice thing about this, because it doesn't have all those long wires, but instead it's just nodes, is that even if you had one of those little chips for each one of the neurons in the brain, it only takes up about 1 cubic centimeter of space, which is about 1/1700th of the size of the brain. I'll just skip that one. Anyway, so computational demands, I'm not going to go into the details of this, but if you calculate how much energy the brain uses and how much it takes for one action potential and how many action potentials occur typically, we can calculate that if you had to translate this to the model with the many components, the compartmental model I was doing for the whole brain emulation, so if you had 10,000 compartments for it, you would need about 1.2 exaflops to be able to do the computations. That sounds a lot right now, but for instance, the Indian government has already just put $2 billion down for building a computer that will run at 132 exaflops in 2017. So that would be fast enough for 100 of those whole brain emulations at the same time. And the price is quickly dropping for this. What I'm trying to say, really, the whole crux of the message is that these are very concrete requirements and projects. The volume microscopy exists and just needs to be scaled up. Molecular ticker tape is coming out in 3, 6, maybe 18 months, depending on whether we're talking prototype or real system, and the chips will also be made eventually. This is just an example showing the real work that's been going on. So for example, on the right-hand side up there, those are chips that have been integrated inside cells and that were functioning while the cells were still alive. Okay, I'll skip that one. And I just wanted to compare quickly, how does this compare with the biological approach if you look at maintenance? On the left, we see what we call the diagram, basically, of all the places where we break down in aging, all the places in the biological system where breakdown happens. This was made by John Ferber. The problem with that is that all of these different connections you see here, they all require different projects to solve them. It's not like you can just have one engineered solution for all of them. So it's a really big problem. And on the right, when you look at the data acquisition approach, well, it's quite a bit more direct and simpler. I'm going to skip all this stuff and just say that, So what we do at Carbon Copies about this is that at the same time we're looking at the big picture, because it's very important to be able to look outside of the box and look at different approaches that are possible. That's how we come across these kinds of projects, how we figure out how to put them together, how to get people working together and talking to each other. But also at this time, because it really is something that's concrete and feasible, it's very important that when we talk about solutions, we need to get down to the details, like what I'm showing you here in that corner on the right. We really need to design and engineer the systems. It's time to do that. And often when you hear people talking, well, it would be really cool to do this, cool to do that, that's nice, but we need actual projects going forward. So thank you very much for listening to me.
Thank you, Randal. Thanks.